The mammary gland is so fascinating, wonderful,
Hmm.
and frustrating because it's so dynamic and it does lots of different things all at the same
Hmm.
time. It's wonderfully frustrating.
Yes.
It,
Good.
it's I like the puzzle and the question.
Welcome back, everyone, to the Dairy Science Digest. This is a podcast designed to bring the Journal of Dairy Science straight to the ears of dairy producers. I'm Reagan Bluel with the University of Missouri Dairy Team, and today's guest is Dr. Benjamin Enger with the Department of Dairy Science at the Ohio State University. And he and his team up in Wooster are dedicated to best understanding what's happening in that mammary gland through different phases of development. But more specifically, today we're going to zoom in and talk about how that mammary gland responds to infection. And chances are, if you're milking cows, you've heard about Staph aureus, that bug that we love to hate. And this contagious pathogen can really wreak havoc on your bulk tank somatic cell count and hide in your herd like none other. So, to better understand this pathway and one of the ways that it can penetrate into your herd, Dr. Enger and his team has been looking at these infections in pregnant heifers prior to freshening to see how that can change the path of profitability for your replacement herd. Their results were summarized in the feature article titled The Impact of Staph aureus Intramammary Infection on Cellular Proliferation and Apoptosis on Developing Mammary Glands of Pregnant Heifers. Oh, that was a mouthful, sir. So, Dr. Enger, welcome to Dairy Science. Thank you so much for joining us today. Could you please take a brief moment to introduce yourself to our listening audience?
Yeah, Reagan, thanks for the invitation. Happy to be here. Exciting to always talk about the research we're working on and continue to scratch our heads on every single day going, well, this is a little different than we expected, but we learn more with every new experiment we do. But my name's Dr. Ben Enger. I'm originally from out west, Idaho, Montana. Got my undergrad at University of Idaho and then did a master's at Washington State with Dr. Larry Fox focused on teat disinfection. Then I decided to switch things up and moved all the way over to Virginia Tech, where I got my PhD focused on mammary physiology and kind of the intersect of how mastitis disrupts mammary tissues and kind of looking at the pathophysiology of why are we losing milk during mastitis cases. And I kind of stumbled upon this area of going, well, where do these intersect really well? And my PhD advisor was an expert in mammary growth. And I was going, well, most of that happens pre-calving before
Yes.
lactation. Where can I look to see if there's something that's worth developing here? And
Yes.
after about probably 8 to 10 years, this is somewhat the culmination of all those different efforts and studies that we've gotten to this point where we actually started working in pregnant dairy heifers.
Yes. And so your team employed 21 Holstein heifers and they were stratified against three different timings of gestation. So about 180 days, 208 days, or 238 days, which corresponds really to 6, 7, 8 months pregnant, and really looking at how infusing Staph aureus into these model critters could impact their production. And as I was reading through this article, I noticed that you also had a kind of a sister article or a paired article with the same group of animals that was published back in November. And guys listening in, I'll definitely have clickable links to both articles. But this was a pretty big project, wasn't it? Can you talk through this project and how long were you working towards this culminating event?
Yeah, so some of the basis for looking at this work was really trying to understand why do animals that calve in with an infection already established, why do they produce less milk? And, my original hypothesis was trying to understand, I think it probably has to do with that mammary growth event pre-calving. So during my PhD, I used a model system where we were looking at non-pregnant dry cows and we stimulated mammary growth with estrogen and progesterone and did a similar style of study.
Mm-hmm.
And so we got some results and data from that, and it was a really clean, this is what's happening outcome.
Mm.
And I went, okay, well, let's look at this in non-pregnant heifers, which was the preceding study before these first two papers we're talking about today. And the outcome of that study, we had non-pregnant heifers, we stimulated growth with really large doses of estradiol and progesterone, and then gave a pathogen to 1/4 to see how that was disrupted. And those results very nicely mirrored what I saw during my PhD. And I went, okay, the next part is to do this in pregnant heifers, the
Mm-hmm.
true population of inference that, especially you all, the dairy producers,
Right.
and working in allied industry, want to know the answer for.
Yes.
And so, coming back to the question, how long did this take? It took a long time. And ultimately, this was so generously funded by the United States Department of Agriculture, which is really the main grant agency that funds basic biological and industry relevant research like this. So that was from the National Institutes of Food and Agriculture, NIFA. So, securing the money and then actually doing the project took a long time. This portion of the grant probably took about 3 years to complete, getting the number of animals that are needed, and then collecting all the tissues and then performing all of the tissue examinations that relate to it, and then the publishing process as well. It's a long haul, so to speak.
Yes. Well, and I try every opportunity I can to highlight the work of land grant universities and how important it is to capture this information and have that background knowledge because, frankly, all the listeners out there, not a single one of them wants to choose to terminate one of their Springer heifers. Right?
Yes.
And that's exactly what happened to these 21 animals with strategy after inoculating them with Staph Aureus. 21 days later, they were harvested so that you could gather up some tissue samples. And these tissue samples were then stained to help you measure and determine, physiological changes that occur in the development of a mammary gland. And you found some interesting things that we've really learned and can grow from as the industry. But before we really get into the exact data results, could you please pause and take a little bit of time to talk about what is mammary epithelial cells and or stromal cells, which are topics that we're going to talk quite a lot about. I want a little bit of background there.
Yeah, that's a great question. I'm really glad you asked this question. And it's something I teach in my classes. So basically, parenchyma or mammary epithelial cells are the moving unit in the mammary gland itself. So mammary epithelial cells, they can be forming the ducts that guide the milk down to the gland cistern and then out through the teat. So it's just a ductal structure, but it's made up of epithelial cells. But once we move up higher into the mammary gland, we're going to hit a single sphere like a hollow tennis ball. And the outside rubber portion of that tennis ball is where there's going to be secretory epithelial cells. Now, generally speaking, being able to differentiate which ones are secretory or ductal in a non-lactating mammary gland actually gets kind of hard because the cells look a little bit more similar because you don't see all the milk components in the secretory cells. And so when we focus on our studies of looking at these tissues, the epithelium is the main thing that's going to be most responsible for how productive is that mammary gland. Now, so that's why we focus on the epithelium, and sometimes that's used interchangeably with the term parenchyma. And parenchyma, by definition, is the functional tissue. So when we talk about the mammary gland, the functional tissue or the milk-producing tissue is the parenchyma. Now, for stroma, lots of people incorrectly think that means it's non-functional. That's not correct. Stroma, the S, you can translate to supportive. It is all the tissue that surrounds that epithelium that helps the epithelium perform its tasks. So it's some level of connective tissue. It's all of the blood vessels that are there because producing lots of milk requires lots of blood. And if you were to take away those structures, the epithelial tissue would fail to actually accomplish its job. So it plays an important role. But when we also think about it, scar tissue fits the bill of actually being classified as stroma. So you want lots and lots of secretory epithelial cells. You want lots of parenchymal tissues. But if there's an overabundance of stroma tissue, you might shortchange the development and growth of that epithelial network as we have that mammary gland grow. So it's a balance.
I would love to see how many dairymen out there listening in are nodding their heads because they've all had that heifer that maybe we missed with fly tags, that maybe had some fly bites or some injuries that occurred post gestation, and they come in maybe 3/4 and you felt that quarter and you can feel what he is saying right now. You can feel that development of the scar tissue that maybe potentially was stromal tissue.
Yeah. So scar tissue is stromal tissue. I mean, it's not going to play a role in the function of the mammary gland. It is supportive. Its role is to help protect further damage. Sometimes it
Mm-hmm.
walls off the bacteria itself and causes an abscess, but it's playing a role in helping the dairy cow. Just from a milk production standpoint, it's not something we like to
Sure.
see lots of or an
Yes, and
unnecessary amount of.
you can almost feel it. But you were doing it
Mhm.
with much more precision by actually harvesting that tissue sample and dyeing it. And so talk to me a little bit about some of your observations as far as the secretory or epithelial cell versus the stromal cell. And was there an effect at how pregnant she was in that process?
Yeah, that's a great question. So, a lot of the examinations that we did in these studies are microscopic examinations, which is a super powerful approach to actually assess tissue structure. So, like how much in a microscopic field of view, how much of the tissue are you seeing? Is it epithelium or is it stroma? And you can get down to the nitty-gritty weeds there, which is super helpful. But the challenge with that is it's very hard to appreciate the total mass of the
Mm-hmm.
udder. Is an udder 5 pounds versus is the udder 15 pounds, whatever it may be. And so microscopically, you can examine the tissues, but if you're looking detailed out there, you might miss the bigger forest. But the challenge with these mastitis studies is there's not a great way to measure the entire forest. You can look at mass of the udder, but if there's a
Mm-hmm.
lot of edema and swelling, you're going to think
Mm-hmm.
that there's more tissue there, but it's just all fluid. So it gets really, really nitty gritty. But back to your point about why are we looking at these late gestation animals? So we euthanize these animals after 3 weeks of infection in each respective group when they were 6.5, 7.5, and 8.5 months pregnant. The reason we zeroed in there is that's when the greatest amount of mammary growth and development actually takes place. So when we look at, a mammary gland and we remove it from a dairy heifer, there's the epithelium and it's growing upwards from the teat towards the abdominal wall. And then surrounding it is kind of this fat pad matrix where there's lots of adipose and it gets eventually replaced by that expanding epithelium. So if you were to think about it, think about like a tree growing upwards into the sky, but maybe a little bit more resemblance
Mm-hmm.
of a bush, so to speak, growing upwards. It's a little bit more dense and not as stringy as just branches going up into the air. And so that process is very important. We want to see good expansion up into
Mm-hmm.
that fat pad matrix. And then by the time we hit full pregnancy and that calving event takes place, in a dairy animal, almost all the
Mm-hmm.
fat is completely gone. And we don't really have an understanding just yet as to what are all the mechanisms that control it, but we do know we want that expansion because that
Milk.
translates to more
Yes.
milk producing tissue. And so we were trying to understand, is an infection interfering with that process? And the answer is yes, kind of. It depends. This was
Yes.
messier than we could have ever
Yes,
possibly imagined.
right. So in your introduction, you were talking about all this clean data, but when I was
Yeah.
reading this paper, it felt less than clean, and sometimes science does that to us. But let's talk a little bit about specifically 6.5 versus 8.5 months. There was some really interesting cellular changes that was occurring in that time. Talk me through, do you believe those changes were a function of just change that occurs biologically to animals as they're gestating and preparing for that subsequent lactation? Or do you think it was really an impact of the infection that was induced for this experiment?
So,
Yeah.
yes to both. Yes, the mammary gland is going to grow and really, really get ready for lactation right in that last trimester. And biologically, that would make sense. That's when we're going to see the bagging up in the, couple of days preceding. But that's when we're also going to see, wow, the mammary gland really has changed. And when you think about that dairy heifer when
Yeah.
she was bred compared to about 6 months pregnant or even 5 months pregnant, you might not see a whole lot of difference. But the concentration of like estrogen and progesterone, which really drives mammary growth and development at this time period, really starts ratcheting up in that last trimester. And that's really what's
Mm-hmm.
pushing this process forward. With the context of the infection on top of that, we see muddled responses where some of these processes of growth and expansion are altered, and then some that we expected to see were
Mm-hmm.
not cleanly altered in the way we were expecting. And it was confusing because we saw all these previous works going, these are clean answers, yes, no. And here the answers were difficult to digest. And what we chalked it up to, which, I have two children of my own, and my wife is probably super annoyed with me,
Yes.
always asking questions of, how's this going for you? What do you think's happening? And she's actually my research associate,
Mm-hmm.
and she has a master's in dairy science as
Nice.
well. So she gets bothered by me at work and at home. But what I think is probably going on here is the reason the data here in this example are different than what we've seen before is we
Mm-hmm.
have the influence of pregnancy. Now, we're going to start dabbling in immunology a little bit, but during pregnancy, there has to be some level of
Has to.
immunomodulation and
Right.
maybe perhaps to say immunotolerance. I don't like the word immunosuppressed because that's not correct. But there's weird things that happen during pregnancy that we wouldn't ever expect to happen other
Yes. Otherwise,
times.
that fetus would be expelled because it's a foreign body, right?
Exactly. So if you have an over robust immune
Mm-hmm.
response, it could put the fetus at risk. So maybe potentially why we're seeing this muddled, messy response in tissue growth, cell death, apoptosis, proliferation, all that is because there is some level of tolerance going. We don't
Mm-hmm.
want to try to burn down the house to fight the infection. We need to simply try to keep it at bay
Mm-hmm.
until the time period is safe to do so. And so that was kind of where we were kind of left with at the end of this study. But we can go forward what that actually means for milk production as we go forward. But um,
Mm-hmm.
the magnitude of changes that we were expecting to see did
Mm-hmm.
not materialize anything close to what we were expecting. We see some responses of, okay, sometimes in this region of the mammary gland, apoptosis goes up, but then when we go to this region of the mammary gland, apoptosis goes down. And it just became a very complex
Yes,
picture to
right. And
digest.
so apoptosis is a fancy word for cell death, right? Or
Yep.
reformatting that area so that it can be productive. And so let's talk a little bit about the difference between the edge tissue sample that you collected
Mhm.
and the one near the center.
Yeah, so basically, as I was discussing how the mammary epithelium grows upward into that fat pad, basically at that edge region is typically where we'd expect to see more growth because that's the tip of the branch, so to speak. If we go back to the tree analogy, that's where it has to grow upward. And so we'd expect to see more growth there, but to a degree, it's also a little interesting because it's actually farther away from the teat, which is where the Staph aureus would come in. And so it's this balancing act. Whereas when I say center mammary tissue, it's really kind of just right near the gland cistern, right in the center basis where that tissue has been grown and developed and it's established. It's not necessarily nearly as dynamic where we have expansion into that fatty tissue. It is already mammary tissue that is potentially capable of maybe synthesizing and secreting milk should the cue get turned on saying, go ahead and make milk.
Fascinating. And guys, if you have a minute, page 4 has some really cool slide pictures that are color, and you can really identify the differences between the cell. Then, additionally, in the November article, you also have some stained pictures. You can visually see the differences between these mammary glands, and you can visually see the impact of these disease causing agents wreaking havoc in your money making tool here. So talk to me a little bit about the rearrangement of these cells and what the duration of impact is. This going to just impact that first lactation for these heifers, or is this going to have a longer ripple?
That is an excellent, very, very loaded questions. And it's something I don't know the answer to. The
Right.
most correct answer I've been told is it depends, and that it
Yeah.
just applies to any question whatsoever. Really, the infection is mostly influenced by the host response to the agent. So we all know, most of us are very, very familiar, sadly, painfully familiar with what are we going to experience with a Staph aureus infection versus what are we going to expect
Mm-hmm.
with an E.
Mm-hmm.
coli infection. And so when we start thinking about that, we have some infections that are relatively benign and the cow gets it and it doesn't cause that much of a somatic cell count and things kind of take care of themselves. Whereas if we have really like a long-term chronic infection with Staph aureus
Mm-hmm.
or even maybe Klebsiella or something
Mm-hmm.
of that nature, the potential for this to go off the rails and just continue to have increasing burdens on that mammary gland's ability to develop. Yeah, I expect that to
Mm-hmm.
be probably not recoverable. The reason is because I think it can result in an excess
Mm-hmm.
deposition of scar tissue within the udder itself. And everyone should be rather
Right.
familiar that scars don't really go away, partially because they shouldn't, but it goes to show that it's really hard to erase
Right.
the damage that has been done in that perspective. There possibly could be the rescue to a degree of allowing that cow to get close to her potential, but there probably is permanent
Mm.
reductions where she'll never be
Mm.
quite as
Well,
good would have been
is it
without it.
appropriate for me to nudge and remind everybody here in April, get those fly tags in, right? Of course, you infuse Staph aureus into these heifers for a controlled study, but in the
Mhm.
field, how does Staph aureus get into heifer mammary glands that are pre calving?
Yeah, that's a great question too. So Staph aureus most often is associated or believed to be associated with the fly biting that you're discussing about. So back in, I think it was the 70s and 80s, there was a very productive
Mm.
research group down in LSU, Louisiana State University, at what they called the Hill Farm, which was a dairy research group. And they did tons and tons of mastitis research there. One of the main
Mm-hmm.
people leading that was Dr. Stephen Nickerson. And they did lots of investigations on how is Staph aureus transferred by fly bites, and they have some really exciting studies to look at that. Just really proof of concept of
Mm-hmm.
can the staph be spread by fly bites? And a lot of the data that we have today about using fly control and does it translate to reduced prevalence of Staph aureus mastitis on our lactating cows, and most importantly for their focus,
Mm-hmm.
your pregnant heifers? The answer is a resounding yes. And they even had
Yes.
a feed
Mm-hmm.
additive, Clarifly, which is something that disrupts
Chitin inhibitor.
the basically something in the
Mm-hmm.
manure.
Yeah, disrupting the exoskeleton. It's really fascinating. And at first, as an extension agent, I was very nervous about Clarifly because our heifers are housed out on pasture, right? And it's and dung
Mhm.
beetles. Dung beetles also have exoskeletons, but pretty quickly I was corrected that that particular chitin inhibitor does not impact your dung beetles. So don't worry, it only kills those damaging flies, but still doesn't impact your manure cow pies. But I've gone down a different hole. Rewinding back to what you were saying about it, kind of depends on the response of the heifer, and kind of
Mhm.
focusing in on that November paper a little bit. You talked a lot about an immunological response. Could you frame up what was observed when you were looking at immunological response, somatic cell count, and all of these
Mhm.
things in the November paper that are relevant to this discussion?
Yeah, so I think I want to frame this discussion a little bit as to this idea that maybe there is some immunotolerance that might be taking place during pregnancy. So when we started taking these animals, we see a
Mm-hmm.
dramatic increase in the somatic cell count, which is exactly what you would expect. Yes, you can go out and very delicately express some of the mammary secretion that would be in those young, delicate teats into tubes, and we can, in the lab setting, count the somatic cells, and then we can actually start
Mm-hmm.
looking at what are the immune cells that are present. So neutrophils, macrophages, lymphocytes. So that would
Mm-hmm.
be an example of like a B and T cell, and trying to break down what that is. And overall, the pattern is
Yeah.
exactly what I would expect to see in even both lactating and non-lactating dairy cows. Lots of neutrophils, the main cell that shows up to fight a new infection. And then not as many macrophages are going to be present because they kind of get displaced a little bit. I'm not saying that there's fewer
Mm-hmm.
total number, but a relative percentage of the whole. They're just not occupying the same amount. And then a lot of B and T cells show up because they're needed there to
Yeah.
start looking at what should they be doing. So we see that, but what was kind of interesting is we generally didn't see this very robust clinical response. Maybe
Mm-hmm.
we saw it for a day or two after we immediately put it in. But overall, we don't see much other clinical signs of disease, which is half the reason these infections are so challenging
Yes.
to prevent on farm because you can't see them. And that's pretty typical of what we would expect when we start contrasting lactating mammary glands versus non-lactating. Well, there's several studies out there where they infuse even E. coli into the udders of dry cows, and
Wow.
no clinical signs present. But then once that mammary gland turns on, the bacteria is still in there. It just seemed to be maybe in a little bit of a quiescent state. It doesn't have as much nutrients to grow off of. There's no lactose.
You bring milk in
Yeah.
there,
And
yeah,
then
milk's
everything
got all the
explodes.
sugars, and yes, and it's nice and warm, about 101.5. It's just perfect for bacteria to flourish.
Exactly. So from an
Yes.
immunological perspective, we see the things that we're expecting to see in terms of cell numbers, cell counts, but the severity of disease, which is going to be really the cow's
Hmm.
response to what she sees, is much more subtle than what I was expecting to see in these particular pregnant heifers.
Yeah, just a function of perhaps where she's at physiologically. And
Yeah.
wow. And so lots of really good, insightful information here. Let's summarize it all up. What are a few take home pieces and parts that you really think boots on the ground dairymen need to know from this long term, multi year project?
So I think I have a couple of good take home messages here. I think the first one is things are a lot messier when you're talking about a pregnant heifer's mammary gland. The immunological responses are different. And so we're not going to necessarily see always the same response of cell death and then cell growth and proliferation. It's going to be influenced by the individual animal. But what I would say is based on all these data I've seen, and in particular in this study where we see a much more muddied and subtle response, I think it probably comes down to that initial when does the mammary gland turn on? And if these infections are present at that time period,
Mm-hmm.
there is likely these infections probably play a little bit more importance to preventing the mammary gland from turning on correctly and as effectively as we'd like to see versus the pre-calving damage to the mammary gland. Both can take place. That's
Mm-hmm.
don't mistake what I'm saying. But if I had to pick one of what I think there is opportunity to really get at, I think that's really where it's coming from. And the reason I make that statement is there's been several studies out of Louisiana State and University of Tennessee where they have experimentally infused antibiotics to these animals only 2 weeks prior to calving.
Mm-hmm.
And what they can see is maybe about a 10% increase in milk for the first 150 days in the animals that receive it. And my takeaway from that is really we're probably not rescuing mammary growth in the last 2 weeks. But
Mm-mm.
what we may be doing is actually allowing the mammary gland to turn on to the potential it needs. And it's really just can those cells turn on and start secreting milk? And maybe the amount of tissue that's there isn't the biggest limiting factor in this specific context.
Yeah.
So even though they did this experimentally and put in antibiotics prepartum,
Mm-hmm.
I just want to emphasize to everyone that that is off-label drug usage. This was done for an experiment to understand dynamics. If this is something you ever want to explore, this is off-label drug usage and it requires a veterinary prescription and obviously a suitable relationship with your veterinarian for proper concerns of is this appropriate? Is it going to likely benefit your farm? As well as there's the likelihood of making sure your residue concerns are properly mitigated and managed because
For sure.
antibiotic usage always increases the risk of antibiotic residues. It's just as simple as that. So that's kind of where I'm at with that. The other thing I'd add there is from a dairy producer standpoint, really examining your somatic cell counts at that time of first test is going to be
Mm-hmm.
helpful because one of the biggest challenges with this is as and the reality is you can't miss what you never had.
Mm-hmm.
And it's really hard to say, I have a problem with heifer mastitis because my milk yield is here,
Mm-hmm.
but you don't know where it was ever supposed to be.
Could be.
So that's the challenge with dealing with these infections is going, where is the baseline? And we have baseline estimates and rules of thumb, but sometimes those are more associated with normal heifer growth. Is she at the right mature size?
Mm-hmm.
And so it gets a little muddied there, but that's an important point I just want to point out. You never miss what you didn't have. And if you didn't know you were supposed to have it, it's really hard to improve.
Really emphasizing that first milk test on your first calf heifers and study your data, guys. I mean, kind of circling back, you got to have the data there. So call your milk tester
Mm-hmm.
out. Make sure you have invited them every month so that you have long-term data to analyze. But flip through those records. See where your first calf heifers are freshening in. Is this becoming an issue? Because if that organism decides to hang out in the host of your replacement heifer at say 6 months of gestation and bring it into your barn, she is a source of contagious pathogens for the rest of the herd too, right? And so
Mm-hmm.
just one misstep in heifer development can directly impact your entire facility and productivity. It sounds extreme, but it's real. These little details really matter. Well, Dr. Enger, this has been a great discussion. Thank you so much for your time and your willingness to share this deep dive in mammary gland development. I know that this will help producers better visualize what's happening to those late gestation heifers as they're coming into the barn. And listeners, I applaud you for taking time out of your day today to better understand Staph Aureus and how it might infiltrate into your herd and have lasting impacts on that mammary gland development. I've really enjoyed this discussion. I've learned a lot from our conversation. This has been the April edition of the Dairy Science Digest, which is a monthly podcast project designed to bring the Journal of Dairy Science straight to the ears of dairy producers. Because we highlight peer-reviewed research articles actively in press, because it's sound science to base your management decisions around and provided to you by your University of Missouri dairy team. So please be sure to like, share, and subscribe to get future editions straight to your cell phone. This is Reagan Bluel with the Dairy Science Digest, and I hope you have a great day.